So we'll begin with the male reproductive system, and we're going to look at it as external genital organs, where you have scrotum with the testes, which produces sperms, the male gametes, and testosterone. And then it passes through a duct system. Part of the duct system is included in something called the spermatic cord. And then we'll see how that duct system finally communicates with the urethra, and then the urethra passes through the penis, and that's how the male gametes are finally come out of the body, okay? And all of these other accessory structures help in nutrition for the sperm and, to keep the sperms motile. So we'll be kind of looking at under all of these various topics. So let's first look at the scrotum. The skin of the scrotum is extremely crinkled, and that's what is meant by rugose skin, very, very crinkled. It contains the testes and the associated ducts with it, and we'll see that there is a duct called the epididymis, and then there is another duct called the vas deferens, which passes out, and this is where it is passing out with blood vessels and nerves, and this area is what is known as the spermatic cord, as you can see here. The scrotum, it has a septum, so the two testes are separated from each other so that they don't kind of, bang against each other and cause any bruising. There's a smooth muscle which is present in the scrotum. This is supplied by the sympathetic nervous system. That smooth muscle is called dartus muscle, and this is important for thermoregulation, because the testes need to be outside the body in order to be functional. That's why they are present outside the body. And if it is too hot, this dartus muscle helps to dissipate heat. If it's too cold, the muscle kind of helps to conserve heat. So that's how it helps in thermoregulation, a very, very important muscle present within the scrotal bag. Then when we look at the testes, as I said, it needs a lower body temperature to be functional. In fact, the testes actually develops in the posterior abdominal wall, and the fetus travels all the way down into the scrotum. Sometimes the testes does not descend into the scrotum when it is known as undescended testes, or this condition is also known as cryptorchidism. Cryptorchidism. Orchid is something that is used for testes. So if the testes is inflamed, for example, we call it orchitis. The importance of this is that if the testes does not descend and stays within the abdominal cavity in a little canal called the inguinal canal, then the testes will become non-functional. It won't be able to produce sperms. And the other important thing is that an undescended testes is also prone to cancer. So with the result, when a child is born with undescended testes, they wait for just a few months to make sure that, the testes is not going to come down naturally. If it doesn't, then as soon as possible, they like to bring the testes down to prevent, one, cancer, and second, the testes becomes non-functional because a high temperature kills off the cells. Now, we look at all of these, so you can read all this part up here. We'll be looking at how the testes is divided into lobules. Within each lobule, you have these little ducts called seminiferous tubules, which actually have the germinal epithelium, which will finally give rise to spermatozoa. And then that has to pass out from the testes in order to finally reach the penis. So we'll see that there is a duct called the epididymis where these are stored and they mature. And we'll also be looking at these other cells which produce testosterone. So let's look at all of these. So here, when we take a cross-section of the testes is actually surrounded by a sac which is continuous with the primitive peritoneal sac. So in fetal life, if this is the abdominal cavity and somewhere here is the scrotum, the peritoneal cavity, which belongs to the abdominal cavity itself, comes down into the scrotum like this, and the testes develops over here. So this kind of acts like a little cavity for the testes. Soon at birth or even before birth, this part closes off. So all this is closed off. So that's why the abdominal cavity, the peritoneum there is separate, And this thing, which now has no connection with this, remains surrounding the testes. So this part is what is called tunica vaginalis. It's very similar to a pleural cavity or a peritoneal or pericardial, where, remember, the organ just invaginated around it, right? So you have a visceral layer of this tunica vaginalis. You have a parietal layer of the tunica vaginalis. In between the two is little fluid which lubricates the testes. Then inside, the testes has a very thick capsule. The capsule is called tunica albuginea. Albuginea means white in color. So here is the thick capsule. And this capsule sends in septate, dividing the testes into smaller areas which are known as lobules. Inside each lobule, you have about 2 to 4 highly coiled ducts, which are known as seminiferous tubules. They are little tubes inside. If you were to cut open a tube, this is what a part of the tube would look like. When you cut open the tube, inside the tube, you'll have a layer of cells, starting from the bottom, going all the way to the top. This layer, from the bottom layer has stem cells, which are known as spermatogonia, and we'll see that when we look at spermatogenesis. This spermatogonia then will divide and will give rise to spermatocytes, which will divide and give rise to secondary spermatocytes, and finally you'll have sperms, which will be in the lumen up here. So that's how in the seminiferous tubule sperms are produced. Sperms need some nutrition. So to help with nutrition, in between these cells of the germinal epithelium, you have special cells which are present, which are known as sustentacular cells. And up here, this one here, these are called sustentacular cells, or also known as Sertoli cells. So these are present, and what these do is, imagine a cell present here, they provide nutrition. The sperms kind of adhere to them, and they provide nutrition to the sperms, these sustentacular cells. Then later we'll see they also secrete other hormones. In between this coiled tubule, so it's highly coiled, so you'll get little sort of round cut sections of them. In between the coiled tubules, you will find cells which are present, which are known as Leydig cells. So in between the tubules, somewhere here, you would find these Leydig cells, which are called interstitial cells, because you can see they are present in between. And they produce testosterone, which is important for maintaining secondary sexual characteristics. It's important for spermatogenesis and various other functions. Now, traveling from, so all inside these seminiferous tubules, sperms are produced. These sperms then travel out of the seminiferous tubules, and you can see that traveling out, they kind of form a little network up here. That network is known as rete testes. And then they connect by means of numerous ducts, which are known as efferent ductules, to a highly coiled duct present here, which is known as the epididymis. So the epididymis is a very, very large duct, but it's so coiled that it kind of coils upon itself, forming this comma-shaped structure. So the epididymis, we'll see, has a head, it has a body, and it has a tail. It's one duct only. It looks like there are many ducts here, but it's actually just one duct, which is highly coiled. The tail of the epididymis then leads into another duct, which is, the name just changes. It's called the vas deferens. And this vas deferens then passes out into a structure which has connective tissue around it, and that structure is called the spermatic cord. So along with connective tissue and blood vessels, you have this vas deferens passing out. So this will now go back into the abdominal cavity, and we'll kind of trace its path, okay? So sperms from here pass into the epididymis, then come into the vas deferens, and then we'll follow that along. So let's answer a few questions. What is the function of the tunica vaginalis? Yes, it's for lubricating the testes. It's outside. As I said, it is very similar to the pleural, peritoneal, and pericardial. In fact, it's part of the peritoneal cavity. It just lubricates the testes. So we leave the testes and its ducts, and then we come to the other part of the external genitalia, which is the penis, male copulatory organ. It has 3 parallel cylindrical structures which are made up of highly vascular tissue with a lot of spaces. Anything which is very vascular with a lot of spaces which can fill with blood is known as cavernous tissue. So these are three cylindrical cavernous structures. We'll see that there are two of them called corpora cavernosa. A single one is called corpus cavernosum, and one which is called corpus spongiosum, which has the urethra. That's why that part of the urethra is often called the spongy urethra. And the skin of the penis is extremely delicate. It can be sort of pulled over the glans penis, and when we remove that, that's what is removed in circumcision. When it kind of goes over, that's what forms the prepuce. So let's look at these three structures in the penis. So here's how the penis begins. What you see is only this part of the penis on the outside. It's only this part which is seen. This part here, which is the root of the penis, is kind of attached to a membrane which stretches between the ischiopubic rami. So there's a central part which is called the bulb of the penis. This bulb goes forwards and becomes the corpus spongiosum. So you can see the bulb is kind of attached to a membrane. So imagine there was a membrane deep behind over here like this, and the bulb is attached to that membrane. It'll go forwards. So here's the bulb, and imagine the membrane up there. It'll go forward and become here is the bulb. It'll go forward and become the corpus spongiosum, and at its distal end, this corpus spongiosum expands and forms what is known as the glans penis. On either side, you have two crura. A single one is called the crus. So these two crura will go forward and become the corpus cavernosum. A single one is called corpus cavernosa. The two together are called corpora cavernosa. And so on either side, you can see this is the crura. On either side, they follow and they come and they go forward as becoming corpus cavernosum, and they insert into the glans penis. Now, if you took a cut section at this part here, this is what you'll see, very similar to this. So you will see the corpus spongiosum with the urethra in it, and you'll see the corpora cavernosa. You can see all these little vascular spaces which are present, which get filled with blood when the penis becomes erect. And there is less erectile tissue in the bulb and the corpus spongiosum because you have the urethra here. If you had too much erectile tissue, what would it do? It would compress the urethra and prevent passage of urine and sperms in that. Okay? The skin of the penis, when you see the skin of this penis can actually be pulled forward like that. And this part which comes forward, this is what is known as the prepuce, or what is also called foreskin. And it is circumcised. Some religions kind of have them circumcised real early in life. Other people do it much later. The idea is actually for health reasons. It's a very good thing to get it circumcised, because when the foreskin is pulled forward, you can see there's a little area present here. Secretions which come out of the urethra can gather here, and they can cause infection. So when you remove that, what happens is that the glans penis is always kind of exposed. So there is no pocket which is present here on either side, and it helps to maintain good hygiene. So that's one of the good things that it's done during circumcision. So let's answer this question. So if the penile urethra were narrowed, what would be affected? And remember, the penile urethra is also called the spongy urethra because it lies in the corpus spongiosum. Okay, this was an obvious question. It's both, right? In the males, the urethra is the only pathway for passage of urine and seminal fluids. So both would be affected. Okay, let's look at the duct system, and I've talked about this already. As I said, you can read all of the stuff. You're not going to go through every part of it. Epididymis, it was a single tube which was highly coiled, and remember it had three parts to it. It's connected to the testes. You have those little ductules called efferent ductules. The epididymis is where someone had asked me a question on why it was so long, and it is because this is a site where you need to store sperm. So many millions of sperms are being produced. You need to store them somewhere, and there's very little space, so that's why it's highly coiled. So this is a site where sperms are stored, they mature. When I say maturation, they actually become motile. When they come out of the testes, they're not motile there. They undergo motility, but the motility is still not complete. Complete motility is acquired, or absolute motility is acquired only in the female genital tract, completely motile in the female genital tract. Which is why when they do assisted reproduction, you may have heard of situations where they do what is called in vitro fertilization. They do that outside of the body. They actually have to put in some sort of an activating fluid to make the sperms absolutely motile. It also produces, in the epididymis, you have deferens being produced, and the epididymis leads into the vas deferens, which is, again, a little bit of storage occurs there, and this helps to transport the sperms. It has a very thick muscular wall, so by peristalsis, the sperms are carried all the way from the testes into the pelvic cavity, which we will see. And then this vas deferens, which is also another name for vas deferens, is ductus deferens. And this is what is ligated or tied off in vasectomy, which is a method of permanent sterilization. So this is permanent sterilization. And then we will look at the other ducts, which are ejaculatory duct and the final pathway, which is the urethra. So, yes?
But you said in a vasectomy that the seminal fluid amount is not any different, it just doesn't have sperm.
That's right. We'll come later to it, where you'll see the seminal fluid. The majority of the seminal fluid is produced by the prostate gland and the seminal vesicles. With this, the testes, the sperm part is only 5%. So when you do a vasectomy, only 5% is affected. So the amount doesn't get affected at all. Yes.
And when you said the absolute motility is acquired in like the female genital tract, is there something in there that activates it? Like, is there like a...
Yes, in the female genital tract, they get a reaction, maybe the acidic pH in the tract. They have a reaction where the sperms sort of become a little bit more motile. They have a reaction where the acrosomal cap and other enzymes kind of change a little bit. So that is done in the female genital tract, yes. There must be the alteration in the pH which does that. So here let's take a look at everything that we've looked so far. We can't see the ejaculatory ducts up here, but here you can see we went over the testes, and the seminiferous tubules lead into these efferent ductules, which then lead into the epididymis, which, as I told you, was one tube. It looks like there are many, but it's one very, very highly coiled tube. And it's kind of arranged in a comma-shaped pattern, with this area, which is larger, called the head. This is the body. This becomes the tail, and that leads into the vas deferens, which you can see then passes out and goes into this cord, which will carry it into the abdominal area in a canal called the inguinal canal. And as it's traveling here, it has blood vessels, nerves, lymphatics, all bound by connective tissue. And so this whole structure here is called the spermatic cord. And in fact, when you do a vasectomy, what you're really doing is you cut through all the connective tissue and you really feel for the vas deferens and you ligate that. So let's take a look at this. Okay, there we go.
The primary role of the male reproductive system is to produce and deliver sperm to fertilize an egg in the female. These sperm are produced outside of the body cavity by the testicles, which are located in the scrotum. The scrotum protects and supports the male reproductive organs that are located outside of the body cavity. These include the testicles, epididymis, and vas deferens. The testicles produce about 300 million sperm each day. However, this is possible only if the temperature of the testicle is 93.2 degrees Fahrenheit. The scrotum maintains this temperature by adjusting how closely it holds the testicles to the body. The interior of each testicle consists of multiple lobules containing 2 or 3 seminiferous tubules. It is within these tubules that sperm formation begins. Sperm moves out of the tubules into the epididymis and then into the vas deferens. The vas deferens carry the sperm upward into the body cavity. After the sperm leaves the vas deferens through the ejaculatory ducts, they pass the openings of the seminal vesicles. The seminal vesicles, which are located near the base of the ejaculatory ducts, secrete a thick yellow substance to nourish the sperm. This secretion forms 60% of the volume of the semen. Semen is the sperm-containing fluid that is ejaculated at the climax of male sexual excitement. From here, the semen travels into the 8-inch long urethra, which transports both urine and semen, but not at the same time. At first, the urethra is surrounded by the prostate gland, and then it is surrounded and protected externally by the penis that is relaxed except during sexual excitement. During sexual excitement, the penis stiffens and enlarges so it can deliver the sperm into the female vagina. During the expelling of the semen, which is known as ejaculation, the prostate gland secretes a thick alkaline fluid that increases the ability of the sperm to move in the semen. Located just below the prostate gland are the bulbal urethral glands. These glands open into the urethra, and during sexual arousal, they secrete the pre-ejaculate fluid that flushes out any residual urine from the urethra. It also lubricates the urethra to help the sperm pass through. When the male ejaculates into the vagina of the female, the sperm must travel upward through the uterus to fertilize the egg in the fallopian tube.
So you saw all the ducts so far, so let's take a look at this question. What is the correct pathway for passage of sperms? Very good. It is seminiferous tubules to the efferent ductules, then the epididymis and the vas deferens. The tunica vaginalis is outside. It's just a little sac which covers it, so it never comes into play. And you saw in that video as well, it never showed the tunica vaginalis. This is just like the, pleural cavity, peritoneal cavity, pericardial cavity, which is just surrounding the testes in order to lubricate it. So we saw the vas deferens. Now let's follow that vas deferens. So in the previous picture, you notice how the vas deferens, or in the video, it showed how the vas deferens went from the testes all the way up, passed through the abdominal cavity, and then it gets into the pelvis. So here we are looking at the posterior aspect of the bladder, and at the neck of so this area here is the bladder, this part here is the bladder, and here's the prostate gland which surrounds the neck of the bladder. And from the bladder, the urethra passes down. So here's the prostatic part of the urethra. So this is the vas deferens, which has come from the scrotum, traveled all the way, and now it's come to the posterior aspect of the bladder, where this vas deferens swells a little bit, and that swelling is known as the ampulla of the vas deferens. Sitting right next to it is again a highly coiled gland, which is called the seminal vesicle. So both of these parts are related to the posterior aspect of the bladder. So here we are looking at the posterior aspect of the bladder. The vas deferens joins the duct of the seminal vesicle. So these two join together and they form a separate duct, which is known as the ejaculatory duct. So you have one on either side. The ejaculatory duct will pass through the prostate gland, and here you can see the prostate gland has kind of been scooped out a little bit to show you the ejaculatory duct inside it. So they pass through the prostate gland to open into the prostatic urethra, because the idea is to get the fluid into the urethra. So this is the only way they can do that. The vas deferens is bringing sperms. The seminal vesicle, as you heard in that video, produces 60% of the seminal fluid. So along with the prostate gland, which we will see in the next slide, the prostate gland also produces fluid. So along with the prostate gland, the seminal vesicle produces the majority of the seminal fluid, 95%. Inside the seminal vesicle, the fluid is alkaline. It has fructose in it, which acts as nutrition. There is mucus, prostaglandins. There is an enzyme also present, which kind of keeps the, when semen comes out, it tends to coagulate, and also enzymes which enhance sperm motility. Then we come to the prostate gland. As I said, it is present just below the neck of the bladder, surrounds the prostatic urethra, has a very, very thick capsule, has some smooth muscle inside it apart from glandular tissue. The glandular tissue produces along, some of the seminal fluid, and as I said, along with the seminal vesicle is responsible for majority of the seminal fluid. So by means of many ducts, it opens into the prostatic urethra. There's not just one duct from the prostate gland. Many ducts open into it. This produces acidic secretions, and it produces a protein which is known as prostate-specific antigen. PSA stands for prostate-specific antigen, and this is what is tested in males above the age of around 50. You like to test the levels of PSA because it is an indicator of how much prostatic tissue there is. If prostatic tissue hypertrophies, the amount of PSA will increase. Hypertrophy means growing in size, could be benign in nature, and that is known as benign prostatic hypertrophy. The levels of PSA will be high. But in cancerous situations, the levels are very, very high. So if someone's PSA is much higher than normal value, then you begin to suspect that it could be cancerous. And of course, you've got to kind of do other examinations you might have to do a biopsy to confirm that. Since in the male, the prostate, the bladder lie anterior to the rectum, by a per-rectal examination, you can actually palpate the prostate gland. Normally, the prostate gland should just feel firm. It should feel like the tip of your nose. In benign prostatic hypertrophy, it will just be much larger, so you won't be able to reach the top of the prostate gland. But in carcinoma, it becomes really, really hard, like rock hard. So then even just by a general examination, again, you can tell whether it is a carcinoma or a benign hypertrophy. In that video, it also showed you this little gland called the bulbourethral gland. We'll take a look at this in the next slide. It's called the Cowper's gland, and it produces an alkaline fluid which kind of removes the traces of residual urine, since the urethra is the passageway for both. And also helps to lubricate the urethra so that the seminal fluid and the sperms can swim along easily. So let's take a look at these these structures. So here you can see the vas deferens traveling from the scrotum along the spermatic cord. Then here at this part, it's passing through a little canal in the abdominal wall, which is called the inguinal canal. And then it gets into the pelvis, where you can see it's related to the posterior aspect of the bladder. This is the swelling, seminal vesicle lying lateral to it. The two join to form the ejaculatory duct. Notice how the ejaculatory duct passes through the prostate gland. Here is the prostate, which is surrounding the prostatic urethra, and by means of many ducts, so many ducts from the glands open from this gland, many ducts will open into the prostatic urethra. So here you have sperms, seminal fluid from the seminal vesicle, and now more seminal fluid which is added by the prostate gland. This then travels through the membranous urethra and comes into the penile or spongy urethra. And here we have the bulbourethral gland, which through its duct opens, as you can see, into this penile urethra. So it pours its alkaline secretion. So this goes out first and washes off traces of urine and keeps the passageway lubricated, so that now the seminal fluid which contains sperms can travel out clearly. During the process of sexual activity, three things occur. The first is just the stimulation makes these cavernous tissue, these three tubes of cavernous tissue, highly turgid, and that is known as erection, and that is under parasympathetic control. So the arteries are relaxed, blood flows into those vascular spaces, and because blood is flowing into those vascular spaces, veins which are on the outside, so if we take a section like this, veins which are on the outside, these veins get compressed. So veins are present on the outside like this, and because this is enlarging, they compress the vein. So this becomes like a closed space. Blood is going in but cannot go out. So that makes it turgid and erect. The second phase is under sympathetic control, which is emission. So what sympathetic nervous system does is it closes off the sphincter, the internal urethral sphincter, because you do not want urine to be passing out at the same time as seminal fluid. So it closes the internal sphincter and it causes contraction of the musculature of all these ducts. So, you know, the seminal fluid travels all that way. And then the final phase is mainly under somatic control, which is ejaculation, where the fluid comes out through the external orifice. And this is because the somatic nervous system acts on those, there are skeletal muscles which surround the beginning of the bulb of the penis and the crura. Remember, the bulb went on to become corpus spongiosum and the crura went on to become corpus cavernosa. And these, the bulb and the crura, are surrounded by skeletal muscles. Somatic nervous system activates those skeletal muscles. They contract, so they squeeze these turgid structures, so that helps to actually milk the urethra and pass the urine out. So let's take a look at this.
The organs of the male reproductive system include the testes.
So we're going to review it again.
Epididymis, ducts, urethra, and penis. The seminal vesicle and prostate and ovourethral glands produce nourishing fluid for the sperm. The testes subdivide into lobules that contain seminiferous tubules that produce sperm. Sperm moves from the seminiferous tubules to the effluent tubules to the epididymis, where they mature. The end of the epididymis forms the ductus deferens, or vas deferens. This structure is found in the posterior border of the testes, penetrates the inguinal, enters the pelvic cavity, and moves over and around the urinary bladder. The seminal vesicles are glands at the base of the bladder that produce seminal fluid. Together with the ductus deferens, they form the ejaculatory ducts. During ejaculation, these ducts eject sperm into the male urethra, common passageway for urine and sperm. The prostate gland surrounds the prostatic urethra just below the bladder. The fluid it secretes goes through small ducts into the prostatic urethra. The vulval urethral glands produce alkaline mucus secretions that empty into the urethra. The urethra passes through the penis carrying semen, a mixture of sperm and secretions of the glands. During ejaculation, the smooth muscle sphincter at the base of the bladder closes off to ensure that no urine is expelled and that semen does not enter the urinary bladder. Sperm moves from the seminiferous tubules to the epididymis, the ductus deferens, to the ejaculatory duct, to the outside of the body by the urethra.
So now you should be very clear on the passageway. Yes.
When you say seminal fluid, does it include any, because only, as you know, only sperm includes DNA, but what's the seminal fluid include? Only ejaculation, or?
The seminal fluid includes fluid which is fluid in nature, which contains prostaglandin, contains mucus,
But
contains...
no DNA.
No, only the DNA is only in the sperm. Yes, it's only. This is just. It's like think of plasma in which cells are suspended. So think of the seminal fluid in which sperms are suspended. Okay, and the entire semen includes that fluid with the sperms. Okay. So let's see from the vas deferens what would be the continued passage of sperms. Oops, okay. The correct answer is number 2. From the vas deferens, you saw it went into the ejaculatory duct, then the prostatic urethra, and then the penile urethra. The the sperms, I'm talking about this passage of sperms. The sperms don't go from the vas deferens into the seminal vesicle, and then somehow from the seminal vesicle into the prostate, and then the spongy urethra. And neither do they, and this one is highly improbable. They can't go from the vas into the prostatic urethra, then retrace their thing and go into the seminal vesicle and come back out into the ejaculatory duct. So if you take a look, that's why I show you these videos. So if you take a look up here, so here is the sperms. They travel this way. They get into the vas, the dilated part of the vas deferens, which is the ampulla. They pass down into the ejaculatory duct. The seminal vesicle pours its secretions into it. So this, those who answered of it going back here like this, that's wrong. Sperms do not get back into the seminal vesicle. They just pass into the ejaculatory duct, into the prostatic urethra and out. The other answer of where they were coming into the prostatic urethra, then going back into the seminal vesicle, and then somehow making their way down here again, as you can see, was highly improbable, okay? So make sure you kind of draw this duct system so you're very clear on how the passage of, I'm talking about sperms, comes right out. Okay, what happens to the internal urinary meatus when sperms enter the prostatic urethra? It closes, yes. If it remained open, urine also will pass out along with sperm. So it closes, and that's what that video showed you, and that's why I played that video to see that. What do you think would be the result of performing a vasectomy, where you ligate the vas deferens? Okay, the correct answer is number 5. Impotence is a situation where the person cannot get or maintain an erection. That doesn't happen here because all you're doing is ligating the vas deferens. You're not interfering with the parasympathetic system at all. Sperms would still be produced. The testes is still going to produce sperms. All you've done is you've just interrupted the transport mechanism. So sperms would continue to be produced. They would travel till the point where you ligate it, and then they would die. And then, you know, they would just be absorbed there. Okay? So the sperms would still be produced. Testosterone is produced by the Leydig cells. You didn't do anything to them. Testosterone continues to be produced. So this will also happen. It's just that the volume would not be drastically affected because I told you the majority of the volume of seminal fluid is by the prostate and seminal vesicles. 95%, sperms only contribute to 5%, so just 5% is missing. So, you know, the person wouldn't notice the difference. So these are characteristics of seminal fluid. It's about the ejaculate is about a teaspoonful. 5 ml is a teaspoon. Normal sperm count is 50 to 100 million per ml. 60%, in order for it to be considered normal, 60% of the sperms should be motile. And why the limit has been set to 60? It isn't that every sperm that is formed is functional. That's why they form so many sperms, because a lot of them are not functional. So there may be sperms which may be double-headed with a tiny tail like this, not functional, plus they've got 2 heads, so they've got double DNA. Or there may be a sperm which has no tail, you know, the sperm may be there may be some other problem with the sperm, okay? So I can't think of any right off the top of my head. So only 60%, if 60% are normal morphologically, that means structurally they are like this, the way they should be, and 60% of them are motile, then the person is okay, okay? They're capable of fertilizing, fertilization is possible. If someone's sperm count is low, and when we say low, below 10 million sperms per ml, then we say that person is sterile. And as I said, after vasectomy, the sperms die in the epididymis and along the rest of the passageway, and they just absorb there, but otherwise the seminal amount doesn't change. And it just, the seminal fluid just doesn't contain sperms.
But this dead sperm, doesn't it create anything on the male?
No, it doesn't create any problems for the male. The only time it will create a problem is that when we did the lymphatic system, if you remember, I told you sperms were sequestered antigens where they do not, they have never come in contact with blood. So when you ligate the vas deferens, as the vas deferens is traveling like this, when you ligate the vas deferens, you have to be very, very careful that the ligature is completely closed off the ends of the vas deferens. Because if some of the sperms happen to come out, then they could cause an irritation and they could cause what is known as a sperm granuloma. But otherwise, the sperms are so tiny that the amount which is present here, they just die off. Nothing happens. It doesn't create any problem. There's no backup like urine would back up or something. No problem at all. Okay. Okay, let's take a look at this question. When do you think sperm count is higher? And don't just guess. There is a scientific basis behind this. Think of why is it important for the testes to lie outside the body. It's higher in the winter. Sperms need, the testes need a lower body temperature to be active, right? In summer, it's hot. It's really hot. So even the outside temperature being very hot, sperms, it's good that it's lying outside, but sperm production kind of slows down a little bit. Winter, when the temperature is much cooler, is really good. So which is why statistically, if you see, women tend, because sperm count is higher in the winter, more babies are kind of born around, take 9 months from winter to around that time. And this is also the reason when couples are trying to get pregnant and they always test the males first, because it's very easy to test the males. All you have to do is look at the seminal fluid, see if 60% are motile, the number is good or not. Then because female testing takes a little bit longer and it's a bit more painful and a lot of more discomfort. So if they find that sperm count is low in a male, first they try to sort of see if they can raise that. So one of the things they do is that if males wear very tight clothing, like they wear briefs instead of boxers, they might tell them to wear boxers just to keep the area a bit, not as tight, and more air can go into the genital area and cool that, and that helps. Then sperm counts do tend to go up after that.
If a male was going in to get tested, like in the summer, would it be so, because it's so hot, would it be so low, would it be considered not normal?
No, it doesn't dip. No, it doesn't dip from 50 to 100 ml. It doesn't come. They don't vary that much. They become sterile in summer and then fertile in winter. No. The dip is just a little bit. Okay, let's look at the process now. How are sperms actually formed? So sperms are what are called gametes, and they are produced through a process of reduction division, which is known as meiosis. So in all of our cells, and even in the stem cells of in the testes, the chromosome number is what is called diploid. It is 23 pairs, or we can write it as, we have 46 chromosomes. So we have 44 and XX or XY. In the male, of course, it will be XY. In the female, it's XX. Or we say that 22 of these are known as, 22 pairs are what are known as autosomes. So 22 pairs are called autosomes. And one pair, the last pair, is called the sex chromosomes. So when you form gametes, what you want to do is that in the females, you want to halve that. So you want to make only 23 chromosomes. In the female, you'll get 22 plus X, because this 44 will become 22 and X will be just 1X. So you'll always get, the eggs will always have 22 plus X. But in the males, because males are 44XY, so you can get 22 plus X or 22 plus Y, right? The sperms could have those kind of chromosomes. Now we look at this, you can, read through this. I'm not going to go through this. So the basic purpose of gametogenesis, as we said, is to provide haploid gametes, so that when they meet, then you get a, complete human being. You come back to that 46 chromosomes or 23 pairs. Now, the idea also is that during meiosis, you want there to be a shuffling of genes, because you get half your, out of the 46 chromosomes that you get, 23 came from your father and 23 came from your mother. But then when, the gametes are formed, you want to kind of make sure that there is a change and there's a bit of shuffling going on. Otherwise, every generation would look exactly the same. And the way you can do that is that during the process of interphase, there is duplication of DNA. So imagine if we drew chromosomes as single threads. So this is the DNA in the chromosome. So imagine you draw 46 of these, but I'm only drawing a few of them, okay? And we call a chromosome pair, that's why we call them pairs, because we have 23 pairs. So we have pair number 1, pair number 2, pair number 3. These are identical chromosomes, and identical chromosomes are known as homologous chromosomes. Okay, so identical chromosomes are called homologous chromosomes. So let's say if I drew, this is pair number 1 and this is pair number 2, and they still just consisted of single threads. You have genes which are present on top of them. Genes are responsible for traits in your body, like, having curly hair, brown eyes, freckles, dimples, all of these things. So when they come close to each other, they can actually exchange material, so that when they divide, then you can have some, suppose this was someone with green eyes and curly hair, but this goes here, and say something else comes here, you may have now green eyes with straight hair. So you can kind of change that because of the hopping over and exchanging. Now, instead of having single threads, if you duplicated the DNA like this, then the chances that you can shuffle genes, the combinations, the more things you have, the more combinations can occur. So the combinations can be a lot more. So that's why your DNA duplicates. The chromosome number doesn't duplicate. It's only the amount of DNA in each chromosome. This is still same one, pair 1, but now each chromosome consists of 2 threads instead of 1 thread. And that allows this shuffling to occur. Now, because you duplicated it, what happens is you'll first divide it. You'll get a cell, you'll get 2 cells with now 1 of the pairs. But remember, the DNA had duplicated, so you've got to divide this again. So that's why at the end of meiosis, you get 4 daughter cells, and each daughter cell has a single thread in it, okay, a single chromosome. So let's take a look at this, and you'll understand this better when we kind of go through this video. And this is kind of explaining the same thing. So during interphase, the DNA duplicates. So you will see that, each chromosome consists now of 2 threads instead of 1. So this is pair number 1. The green one, let's say, is from the father. The purple from the mother. This is pair number 2. So they're only showing you 2 pairs. So here, during the process of prophase, these chromosomes come and line up against each other, and they exchange material. And that is what is known as crossing over. And then during metaphase, they come and lie along the equator. You should remember mitosis from ANP1. So they come and lie along the equator. At anaphase, which is the pulling away of chromosomes, so notice these pairs get pulled to the opposite side. And at the end of telophase, you have 2 cells with, now they have only 1 set of the chromosomes. But each chromosome still consists of 2 threads now, so you have to break those threads as well. So that's why this goes through meiosis 2, which is exactly like mitosis. So again, each of these cells now will have the same phases, so that will be called prophase 2, metaphase, they come and line up along the equator, anaphase, they are pulled apart, and telophase, so you end up with 4 cells, so that each cell now consists of a single thread, okay? So let's take a look at this.
The cell prepares for meiosis just as it does for mitosis.
As the DNA condenses, each chromosome finds its homologous counterpart.
So I'm going to stop you. So this is just showing you after the DNA has been duplicated. So initially it was only one red and one blue, but now notice how it has been duplicated, and then you can see how they'll cross over.
Chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
So you can see at the end of meiosis, therefore, you need 4 cells to be formed. Okay, we'll see how that differs in spermatogenesis and in when we do female oogenesis. So spermatogenesis, that's where, remember, the seminiferous tubules had germinal epithelium, where you have all of these stem cells which are present. The stem cells are known as spermatogonia. So a few differences. Spermatogonia divide, and they are so this obviously occurs in the testes. They are able to maintain the primary stem cell line. So spermatogonia divide throughout life. So males are capable of forming new stem cells as long as they live, except that, of course, the process slows down, but they can keep constantly forming new stem cells. Then the second thing is that the spermatogonia then change. They grow in size. No division occurs. They change, and they become what are known as primary spermatocytes. Then we look at the stages, and we'll see that it undergoes meiosis. They get something called secondary spermatocytes, and finally the 4 cells which are formed are known as spermatids. Spermatids have a lot of cytoplasm in them. They are not motile, so they're very heavy. They cannot swim. So this has to change somehow from becoming 4 cells. So you have, imagine 4 cells which look like this. Heavy, no tail, lot of cytoplasm. They have to change somehow. They have to get rid of the cytoplasm and get a tail. So that change, where from spermatid they become spermatozoa, this doesn't undergo any division or anything. It's just a question of losing cytoplasm and gaining a tail. So it's a structural change that happens. That process is known as spermiogenesis. So let's see, since males produce spermatozoa throughout life, so if one batch is spoiled, you know, they'll get another new one after 74 days. So they'll keep forming that. So the age of the father does not matter as much as the age of the mother, because we'll see when we do oogenesis how the mother forms her stem cells before birth and she never forms any stem cell after birth. So she's born with whatever stem cell line she has. So when she is, say, 25 years old, that stem cell is exposed to, let's say, 25 years of pollution and radiation and so on. But let's say when she's 40 years old, her stem cell is 40 years old. So can you understand that how many years of pollution, radiation, environmental changes have that cell has undergone? So it could undergo mutation. So the mother's age becomes more important at conception rather than the father's age. Okay? So let's look at this. So here we have spermatogenesis. So when we start in this is showing you the seminiferous tubule. So this is the basal lamina, and these are all the cells which are formed. So at the base are these primitive stem cells which are called spermatogonia. The spermatogonia have the normal number of 23 pairs of chromosomes. The DNA and the chromosome number, if we say, is 2n. So let's say that the DNA number is 2n. This undergoes growth, this spermatogonia, and they form further spermatogonia, and they end up forming primary spermatocytes. So they're still just growing in size. Primary spermatocytes are the largest cells that you see inside the germinal epithelium. So no division has occurred here. So that's why you can see that 2n and 2n, primary spermatocyte. Primary spermatocyte undergoes this meiotic division. So it undergoes two meiotic divisions, remember? So the first one forms 2 cells, and then each of these divide and you form 4 cells. So this is the spermatids which are formed. Spermatids are a lot of cytoplasm, and actually the tail is very, very small. It's hardly formed. And then this undergoes a morphological change where it gets rid of all this excess cytoplasm, gets a nice proper tail. So this process here is what is known as spermiogenesis. And spermiogenesis has no division. So this is important for you to remember. There's no division occurs here. The division only occurs from when primary spermatocyte gets converted to secondary, and secondary gets converted to early spermatid. Okay? So the chromosome number and the amount of DNA both halve in this. Both become half. And then when we were looking at the testes, you remember we looked at the Sertoli cells, also called sustentacular or sustentocyte, which are nutritive. So the sperms can kind of attach. You can see this is the big cell. Sperms attach to them for nutrition while they're lying in the testes. Then let's move to the female reproductive system, and when we do gametogenesis in the female, you'll see that the stages are pretty similar, but we look for major differences over there, okay? First, let's look at some of the things that we are going to study. We'll look at the gonads, the ovary, which is the counterpart of the male testes, the duct system, the fallopian tubes, uterus, and vagina, and then the external genitalia, which consists of 2 fatty lips on the outside, which are called labia majora. They enclose much thinner lips without hair, which are called labia minora. The labia minora end just as we had, the corpus spongiosum, corpus cavernosum, which began as the bulb and the crura. We have the same things in the female as well, but they're just much smaller. So the glands, for example, you have glans penis, that is the equivalent would be the glans clitoris, which is present there. This labia minora encloses a space, and that space is known as the vestibule. I don't know if it is labeled up here. So that space is called the vestibule, which is this area here. And in the vestibule, you have both the vaginal orifice and the urethral orifice, and they lie so close to each other, which is why urinary infections are very common in the female. In the male, if you remember, there was a gland called the Cowper's gland, which was lubricating, right? The bulbourethral gland. In the female, you have a gland which is called the Bartholin's gland, or also known as the greater vestibular gland. And here you can see this Bartholin's gland. Again, the purpose is to lubricate the vaginal orifice at this point. And then we'll be looking at the mammary gland as well. Okay, so we'll be looking at all these areas. So let me see why I have the arrow. Go back. So ovaries, you can read all of this. One important thing which is highlighted, unlike the males, remember spermatogonia divide into more spermatogonia, and then the rest will grow and become primary spermatocytes. In the female, the stem cells are called oogonia, which begin to divide during fetal life itself, and then they change. They just grow and become primary spermatocytes. So when the girl is born, no stem cells are present. Only primary spermatocytes, primary oocytes, are present. Okay, so she has no stem cells. So that's why these are exposed. The older a woman is when she's pregnant, the more chances that mutations could have occurred. Then when we look at the ovary, it has just like the testes had a cortex and a medulla and numerous. In the testes we had lobules, but in the female we'll see that you have these follicles which surround these oocytes. These are cells which surround the oocytes. So we'll be talking about these cells. These are the cells which secrete estrogen, because we know the ovaries secrete estrogen and progesterone, and we'll see how these cells secrete that. These little primary oocytes, they mature, they undergo oogenesis. But in the male, 4 cells were formed. You know, each spermatogonium gave rise to 4 functional spermatids which changed into sperms. In the female, 4 cells are formed, but only 1 of them is functional. The other 3 are useless. They are known as polar bodies. That one functional cell which is formed in the female is released at ovulation. So during ovulation, this comes out. And then what is left behind, we look at this structure is called the corpus luteum, and the corpus luteum produces a hormone called progesterone. So we'll be looking at these structures in the ovary. So here again you can read this. The function of the ovary is not only to produce these follicles, but the estrogen and progesterone, they don't only have reproductive functions, but they actually protect women until menopause. They protect women from osteoporosis because they prevent osteoclasts from being very active, and they also increase osteoblastic activity. This is the reason why, because at menopause, the hormones have dipped suddenly. In males, the hormones do not dip as suddenly. They kind of plateau very, very slowly like this. In women, there's a drastic dip which comes down, which is why after menopause, women become more susceptible to osteoporosis, because this protective action by progesterone and estrogen has gone. Okay? Estrogen, which is also a reason why after menopause women are more prone to heart attacks, because our blood cholesterol goes up. In males, that sudden dip not being there, they are prone, there's nothing, they don't have anything as drastic as us. So while their incidence of heart attacks doesn't change at any period in life. I mean, if their diet is not good, they are going to be exposed to it throughout. But in us, we have a bit of that protective influence of estrogen and progesterone. Estrogen being the one which helps to lower our blood cholesterol. Yeah.
Do you think that could be like an evolutionary adaptation to dealing with stress?
Could be must be evolutionary, must have something to do with the fact. And the idea of the drastic dip is so that women are not reproducing throughout the life, because reproduction is it's an assault on the body. There's only so much your body can take. So that's why maybe, evolutionary wise, it came back that your reproductive span was only so much, and that dip then kind of happened because your hormones became less. Okay? Read this bit about the fallopian tube, and we'll be looking at the various parts of the fallopian tube, and there's a dilated part. Remember, we looked at the ampulla of the vas deferens. Ampulla is always dilated. So we'll see how fertilization usually occurs in the ampulla. And the epithelium of the fallopian tube has cilia. It's ciliated columnar epithelium. The ovum is a very, very large cell. It's one of the largest cells in the body. So it's very difficult to actually roll the ovum along. So what these cilia do actually is when the ovum is fertilized and becomes a zygote, and the columnar epithelium secretes mucus, the cilia beat towards the uterus. So they set up a current which kind of rolls. So the water current, kind of mucus current, gently kind of rolls the zygote towards the uterus. Now, if at any point the fallopian tube was blocked, you can understand this zygote would be stopped in the passageway, and then you'd end up with something called ectopic pregnancy. So just as a question, how what do you think is the diameter of the fallopian tube? Give me in terms of some things that you use in everyday life, like, you know, a hose or straw or, you know, whatever. So give me in terms of that. What do you think is you think straw? Anybody else? Like a smaller straw, not the McDonald's straw? Okay. Well, I did get somebody told me there was a hose once, as big as a hose. I mean, they obviously didn't, I've never looked into the pelvis. But any other? Straw was one. What else?
A little bit bigger than a toothpick.
Little bit bigger than a toothpick. Okay. Coffee stirrer? Like a coffee stirrer? Okay. You'd be surprised to know that the diameter of the fallopian tube is 2 hairs breadth. 2 of your head hair. 2. Just so narrow. So can you understand that how easy it is for it to get blocked? Okay. And the other thing in the female is the fallopian tube is open into the peritoneal cavity. So from the peritoneal cavity through the fallopian tube, out through the uterus to the outside. So the peritoneal cavity is actually open in the females, and the males it's closed. So it's very easy for infection to travel from below through the vagina into the uterus, into the fallopian tube, and you get what is pelvic inflammatory disease. So it's amazing how so many women actually don't have pelvic inflammatory disease. Yeah.
Is there a way to tell someone has an ectopic pregnancy?
Yes, you can tell when a person has ectopic pregnancy because it can only grow so much. So usually by about 12 weeks, you know, when a woman gets pregnant, after some time, if she gets an ultrasound and they don't see a viable fetus in the uterus, that's one way of telling. Then such women, because the fallopian tube is expanding, she would be, she would complain of pain, but she's still having no periods, which is called amenorrhea. So then, again, you can begin to suspect that. And so those are ways that you can tell, okay? So, and like vasectomy was the thing, that sterilization procedure, we do tubectomy in females. So let's take a look at the ovary and the fallopian tubes. So here, when you look at the ovary, these are the little follicles which are present. So they are those primary oocytes surrounded by cells of the ovary. So these are known as primordial follicles. Then they get a bit larger, you can see. So these cells become, multi-layered, where you start calling them granulosa cells. When they're single-layered, they call them follicle cells. Under the influence, we will look at the reproductive cycle. So under the influence of hormones coming from the pituitary, they begin to grow in size. Many such follicles grow together, but only one kind of wins the race. And these cells, these granulosa cells, these are the ones which secrete. Remember, the ovary secretes estrogen and progesterone. So as these follicles grow, these granulosa cells are the ones which begin to secrete estrogen. After menopause, you can understand that no follicles are growing there, so that's why our estrogen levels fall, because the ovary is no longer producing that. So as I said, many begin to divide and continue to grow, many begin to mature at the same time. One wins the race. That one is known as the Graafian follicle, and it kind of begins to get a little a cavity inside it, fluid-filled cavity, which puts pressure. And then at ovulation, the cavity ruptures and this oocyte is liberated along with a few cells on the outside. What is left behind, these cells which are left behind, they form something known as the corpus luteum. Luteum is a yellow color, and that's because these cells take a pigment which is yellow in color. And now the corpus luteum is what secretes progesterone. So that's how you have both estrogen and progesterone being secreted by the ovaries. If fertilization occurs, the corpus luteum survives. If fertilization doesn't occur, the corpus luteum dies, and then the second cycle starts again. Okay? So this occurs throughout your reproductive period. I'll show you a picture of the fallopian tube, but let's look at the uterus meanwhile. So again, pelvic organ between the bladder and the rectum. We'll be looking at some pouches of peritoneum which separate it from the bladder and from the rectum. It has 3 layers. Very similar if you think of it to the heart. Endocardium, myocardium, and the outer layer was called epicardium, but here you call it endometrium, myometrium, and the outer layer is connective tissue called perimetrium. We'll look at the three parts of the uterus, the fundus, body, and cervix. And the uterus is supported by many ligaments to keep it in place, and it is exposed, it's becoming pregnant, growing bigger, and then coming back. So it needs a lot of support. Sometimes, if due to multiple pregnancies or raised intra-abdominal pressure, these ligaments become very, very lax. So they're no longer able to support the uterus. So the uterus can actually descend from its position, and that is called prolapse of the uterus. We'll be looking at the cavity, and then also the uterus is supplied by the uterine artery. And when we did the urinary system, if you remember, I told you that the uterine artery and the ureters were very closely related. And one always had to take care when you remove the uterus during hysterectomy, because you could, by mistake, catch the ureters with it. So let's take a look at these. And okay, here is the vagina. It's the female copulatory organ, no glands. It depends on the cervix, the secretions from the cervix for lubrication, as well as the secretions of the Bartholin's gland, which was the female counterpart of the bulbourethral gland in the male. We'll see that the posterior wall is much longer than the anterior wall, and actually there is a little space present there, these spaces are called fornices. And we'll notice how the posterior fornix is much deeper than the anterior fornix, and that's usually where seminal fluid is deposited and makes it convenient for the sperms to swim into the uterine cavity. So we'll take a look at all of these structures. So here let's look at the ovary, the fallopian tube, uterus, and vagina. So here is the ovary. Here is the fallopian tube, which you can see is kind of lying over the ovary. And if you look at the various parts of the fallopian tube, beginning medially from the uterus, this narrowest part of the fallopian tube is known as the isthmus. This travels and comes to a dilated part, which is called the ampulla, and that's where fertilization normally occurs. Then it comes to a funnel-shaped area. This funnel-shaped area here is known as the infundibulum. And then that leads to finger-like processes called fimbria. These fimbria are present so that when ovulation occurs, and the ovum is kind of liberated here into the peritoneal cavity, somewhere here, these fimbria can go and actually grab the ovum and suck it in so that it can pass towards the uterus. And that's what I meant. There's a hole present up here, and so this is continuous, as you can see up here with the outside, and this hole is continuous with the peritoneal cavity. So that's what I meant by saying that the peritoneal cavity is open in the females, because this ovum is liberated into the peritoneal cavity. Then when we look at the uterus, again think stomach. You remember in the stomach, the part above the cardiac orifice, the highest part was called the fundus. So here, if we draw a line across the openings of the fallopian tubes, this part above is called the fundus of the uterus. This next part is called the body of the uterus. And this lower area which projects into the vagina, this part here is known as the cervix. The cavity which is present in the uterus, you can see it's a triangular cavity, continuous here with the fallopian tube. Below this cavity of the uterus is continuous with the cavity in the cervix, so this is called the cervical canal. And there are two openings. The cervix opens into the vagina. So this external opening is called the external os. Os means opening. And this upper opening is called internal os. And so the canal is present between the two openings. Here is the vagina. So the vagina is a closed kind of sac which is going around the cervix. So it's got a little space here on either side. These are called lateral fornices. there will be a space behind it, which will be called the posterior fornix, and then there will be a space in front of the cervix, which will be anterior fornix. So imagine the vagina completely surrounds the cervix, which is kind of hanging. So there's this space all around it. So that space is what is called fornix. So here you can see this is called the lateral fornix. The posterior will be behind and the anterior will be in front. We'll see. The purpose is actually one, the posterior fornix mainly has a purpose, but otherwise it is just that that's the way it is. The cervix projects and the vagina goes up around the cervix, kind of, holding it together. We'll see how the posterior fornix has a reason for it to be present.
So if you use a diaphragm, is it covering?
Yes, when you use a diaphragm, what you use is the diaphragm covers the mouth of the cervix. So that now this os is closed, so sperms cannot swim up. That's what the diaphragm does, okay? The uterus is surrounded by, or it is kind of covered over by a curtain, which is called the broad ligament. This is a curtain of peritoneum, which covers the uterus and the fallopian tube. So this curtain is called the broad ligament, which you can see up here. And so it's reflected behind on the bladder, on the rectum, reflected in front on the bladder. Now, in this picture, it looks like the uterus is straight in the pelvis, lying in a straight fashion. It isn't. It actually is kind of bent over itself and bent in relation to the vagina. So let's take a look, and that you can only see in the lateral view. So if you look at the lateral view up here, you can see the relations also. So notice how the bladder in front with the urethra, the vagina is lying behind the urethra and in front of the rectum and anal canal. Notice how the uterus is kind of bent over and it's kind of lying over the bladder up here. So there is an angle that the uterus has with the vagina. Notice this angle like this. So when it's bent forwards, this is known as anteverted. So you can see anteriorly bent. If it was bent backwards, this is how it would be. It would be bent backwards like this, and the cervix would face this way. So it would be backwards. It wouldn't go outside the sacrum. So it would be backwards. So such a thing is called retroverted. Retro is when it's bent backwards. Anti is when it's bent forwards. Usually it's anteverted. Another thing you can see is it not only is there a relationship with the angle of the vagina, the uterus is also bent in relationship. If this is the angle of the uterus, this is the angle of the cervix. Can you see that? There's this angle with the cervix as well, okay? So in other words, this is the cervix and then the uterus is bent forward. So angle of the cervix, angle of the uterus, okay? So this kind of angle between the cervix and the uterus is called flexion. So it's not only bent in relation to the vagina, which is version, but it's bent in relation to itself. This is the ideal position that you can see. Notice in this how the anterior vaginal wall is short. Look at the posterior vaginal wall, which is much longer. Also notice the anterior fornix is very shallow. Look at the posterior fornix, much deeper. Can you see that? See where the cervix is pointing to. The external os is pointing to the posterior fornix. The posterior fornix being deeper, during intercourse, most of the seminal fluid is deposited in the posterior fornix. That's one of the functions of the fornix. So it makes it very easy for sperms who don't ask for directions, makes it very easy for it to swim into the uterus, right? I told you, I told some class that so many million sperms are deposited in the female tract, but only 50 finally make it up to the ovum, okay? So this makes it very conducive to pregnancy. If, on the other hand, the uterus was facing backwards like this, then the cervix faces towards the anterior fornix, and this could be one reason why sometimes couples don't get pregnant, because it makes it very difficult for the sperms to kind of detour and go around like this, okay? Also, up here, what you can see is this peritoneum. Here's the peritoneum, which goes over the bladder, then reflects on the uterus, then reflects on the rectum. So you get these two pouches of peritoneum. So here this pouch of peritoneum is between the bladder and the uterus. This is called vesicouterine pouch. bladder, vesicle, and uterine for uterus. And this pouch of peritoneum between the rectum and the uterus is called recto-uterine pouch, which is also called pouch of Douglas. After the person who, usually whenever you have a name, it's a person who did Douglas. Douglas discovered it. The pouch of Douglas is rather deep, and sometimes when you can see from a per vaginal examination, you can actually feel the cervix, you can feel if someone had a tumor or there was an ectopic pregnancy, like a question up here, the fallopian tube being very heavy will drop here down into the pouch of Douglas. So through a vaginal examination, you can actually feel a heaviness in this pouch of Douglas. Or even if someone had a tumor of the ovary, you could feel that, like a cystic ovary or something. Okay, don't answer this. Let's go back and let's take a look at this video which kind of shows these. So here's the ovum being liberated at ovulation. Travels through the fallopian tube. Gets into the uterus. You can see the layers of the uterus. And here it's showing you an anteverted uterus. Notice the posterior fornix, so deep. And the vaginal cavity. And this is in the vestibular area. So let's take a look at this next question. What are the consequences of inflammation of the fallopian tube? Very good, both 1 and 2. So pelvic inflammatory diseases usually prevent a woman from getting pregnant. If she does get pregnant, ectopic pregnancies become very common, and you have, depending on how bad it is, you have to either remove the tube or just remove a segment and you can re-anastomose. The interesting thing about ovulation is that the ovaries ovulate at alternately. And so even if fallopian tube is taken off from one side, the fallopian tubes, the pelvic cavity is a very, very narrow space. So even though the fallopian tubes are 10 centimeters long, it's not like they're stretched out with their arms like this. They're very close to each other. So the fallopian tube, when the ovum is liberated, or the oocyte it's called, when it's liberated, there's like chemotactic influence which makes the fimbria kind of swim towards the oocyte. So even if, let's say, the right ovary is ovulating, but the fallopian tube on the right side has been removed, the left fallopian tube can actually swim towards it and pick up the ovum and bring it down this side. And that's called transmigration of the ovum. So it really doesn't affect the woman as much. Okay, let's another question. An anteverted, anteflexed uterus will face the sacrum. Is this true or false? So you looked at that picture and I showed you these version. False. Remember, it is facing in front. The anteverted is sitting on top of the bladder, so it is facing the pubic symphysis. A retroverted uterus will face the sacrum. Another question, what would be the path taken by we'll see when we do gametogenesis that it's actually the secondary oocyte which is liberated at ovulation. So what is the path that it takes when it is released from the ovary? Very good. Yes, from the, it's liberated into the peritoneal cavity, taken up by the fimbriated end, going into the infundibulum, then ampulla, then isthmus, and then uterus. It doesn't go, this is backwards. From the ovary, it goes into the cervix, then uterus, then ampulla, so it's actually going to go out, right? And the same up here, so it's this one. External genitalia, not much. I showed you those areas here. The Bartholin's glands are the most important glands up there, which lubricate the vagina. And then when we look at mammary gland, these are actually modified sweat glands. And the mammary gland consists of adipose, which is the major amount which actually gives the breast its size, and glandular tissue. And then there's connective tissue in between, which divides it into lobes. And in each lobe, you get these glandular tissue being drained by a duct, which is called the lactiferous duct, and that opens at the nipple. And just before it opens is a little dilation called the sinus, where milk can be stored. And the duct system increases during puberty due to estrogen, and glandular tissue increase is more during pregnancy. And we saw when we were doing the endocrine system how this was under the influence of prolactin and oxytocin. So this is external genitalia. Just as I explained to you, you have a bulb and then you have the crura, and they join at this clitoris. So it's just a smaller version of what we see in the male. And up here in the mammary gland, it is in the superficial fascia. It lies over the pectoralis major muscle. And then here you can see the many lobes that it has been divided into. So here's all the glandular tissue being drained by these lactiferous ducts, which have these sinuses just before they open at in the nipple. Okay? And this is just a space where milk can be stored. So when we look at oogenesis, certain differences with spermatogenesis. One is that in oogenesis, you can read most of it up here, but one of the important things is that the process actually begins before birth and is completed at puberty. So oogonia, which are the stem cells, very much similar to spermatogonia, they actually begin to do their job. They get transformed into oocytes in fetal life itself, so that when the baby girl is born, there are no stem cells or no oogonia left. Remember, spermatogonia were there all throughout life. So that's one difference. So there are no oogonia present after birth, whereas spermatogonia are present after birth, and they continuously divide and form newer stem cells. So that's one. Then the other one is that these oocytes, they begin their division, and when they get mature during a woman's reproductive cycle, only about 400 of these oocytes really get to mature, because a woman goes through menopause and you need the hormones to get these oocytes to mature. So since the hormone levels fall, only about 400 during a woman's reproductive life, which is say about the range is about 12 to say close to about 50 years, okay? Then we'll see, when we look at this, we'll see that the primary oocyte, just as you had a primary spermatocyte, remember the spermatogonium changed into, it matured or became larger and became a primary spermatocyte. Similarly, the primary oocyte it has a collection of cells, and together they are called the primordial follicle. So they begin their meiotic division in fetal life itself, and then it kind of remains suspended in prophase, and then again it picks up when a girl achieves puberty, which is around anywhere from 10 to 12 years. So here you can see that at puberty, many follicles begin to mature together, but only one sort of wins the race, and that's the one which is liberated at ovulation. So what is liberated at ovulation is something known as secondary oocytes. So remember, at the end of first meiotic division, two cells are formed. So here in the male, there were two cells which formed, and each one of those two cells divided and formed two more. So you ended up with 4 functional spermatids, which then changed and became spermatozoa. Here in the female, at the end of the first meiotic division, only one functional cell is formed, which is called the secondary oocyte, and the other cell is called the first polar body, which has very little cytoplasm, has no function at all, okay? Then the other major difference between spermatogenesis and oogenesis is that the secondary oocyte, it only starts its second meiotic division and gets completed if it is fertilized. So if there's no fertilization, the second meiotic division is not completed, okay? So if there is fertilization, what you will have is that the secondary oocyte will become the ovum, and then you'll end up with 3 polar bodies. So that's how you get 4 cells. So 3 polar bodies and the ovum. So the second meiotic division is only completed if the secondary oocyte is fertilized. The moment it's fertilized, it completes its second meiotic division. Otherwise, it just passes out of the fallopian tube through the uterus and out of the body. So you can see the ovum is there's only 1 functional body, 3 non-functional cells which are formed. In males, you have 4 functional cells formed. In the female, only 1. Because if we had 4 functional cells being formed during every ovulation, imagine the population explosion that's going to be there. Everybody would have quadruplets all the time, right? So here you can see, this is when we look up here. So here's an oogonium, the primary stem cell. It grows and becomes the primary oocyte, just like the spermatogonium became the spermatocyte. This then begins its meiotic division. So during interphase, remember the DNA duplicated so that there could be genes reshuffled. So then it divides into 2 cells. As you can see, they are unequal cells. So this is the first meiotic division you have. The one functional cell is called a secondary oocyte, and then the other non-functional cell is called a polar body. Each one of these undergo meiosis too, so that the DNA also gets halved. So the secondary oocyte then forms the ovum and another polar body, and this polar body forms 2 more polar bodies. So that's how you have 1 functional ovum and 3 polar bodies. This process will occur only if the secondary oocyte is fertilized. If it does not occur, then all this is not formed. Only these will be formed, and they'll just be passed out of the body. Okay? Now, in order for all of this to happen, there are 2 cycles which kind of go on simultaneously. One occurs in the ovary, and in the ovary, the hormones are produced, and the hormones produced are estrogen and progesterone. And during this, in the ovarian cycle, we'll see that many follicles begin to start maturing. So that phase will be known as the follicular phase, because many of the follicles begin to start maturing. And if you remember, when we did the endocrine system, in the anterior pituitary, there was something known as FSH and LH which were produced, follicle, that's why it was called follicle stimulating hormone and luteinizing hormone. Remember, they were under control of the gonadotropic releasing hormones from the anterior hypothalamus. So those two sets of hormones will be acting. So gonadotropic releasing hormone will act on the anterior pituitary, which will produce FSH and LH, and these will cause several follicles to mature. So that phase which occurs is known as the follicular phase, and as I said, one of these follicles kind of is a bit faster than the other, and it kind of matures and grows much faster so that the other follicles begin to die off, and the one that matures will be known as the Graafian follicle or the mature follicle. That Graafian follicle will rupture and the secondary oocyte will be liberated. That process where the secondary oocyte is liberated is known as the ovulatory phase, so we'll take a look at that. And then after ovulation occurs, what is left behind, we will see in the ovary, are those cells which surrounded that, the primary oocyte. Those cells begin to acquire a pigment, and they look a bit yellow in color, so that what is left behind in the ovary is known as the corpus luteum, and so therefore this phase is known as the luteal phase, and this corpus luteum will secrete progesterone. Now the hormones which are secreted by the ovary, estrogen and progesterone, are secreted by those cells which kind of line those oocytes. Those act on the endometrium of the uterus. So they act and cause what we will see is a uterine cycle, okay? So some of the things you've got to remember and not be confused with is what are the phases called in the ovary and what are the phases called in the uterus. And if you think of what happens in the ovary, it should be easy to remember. Where are follicles present in the ovary? So you have a follicular phase in the ovary. That's the beginning. Where does ovulation occur in the ovary? Because that's when the secondary oocyte is liberated. So the ovulatory phase is also in the ovary. And what gets left behind in the ovary? The corpus luteum. So they have the luteal phase. What does the ovary secrete? Estrogen and progesterone. The ovary is under control of which hormones? FSH and LH coming from the anterior pituitary, which in turn is controlled by the anterior hypothalamus. Okay? So here is the chain. This estrogen and progesterone then causes the uterus to undergo phases. The uterus does not produce any hormones. So one thing to remember. So when we have the uterus, what are the things that happen in the uterus? One is the uterine lining gets removed. And when the lining gets removed, that is the time when you have menstruation. The endometrium is removed. So that is known as the menstrual phase. The menstrual phase occurs in the uterus because the endometrium is shed off. So the menstrual phase. Now when the endometrium is shed off, it's got to repair itself and come back again so that it can be shed off over the next menstrual or next cycle, or it has to be repaired so that if pregnancy occurs, it has to be ready to receive the fertilized ovum. So when it begins to repair itself, the first thing that will happen is it will begin to grow in height, right? So it will proliferate. So you have a proliferative phase. And here the proliferative phase, as you can see, is under control of estrogen, which is produced by the ovaries. After the proliferative phase, the endometrium thinks, okay, there is a likelihood that the ovum will be fertilized and I've got to prepare. So I've got to make the entire endometrium very rich and very moist to receive this ovum. So I've got to secrete a mucus and I've got to secrete substances so that, I prepare a bed which would be just ideal for the fertilized ovum. So that phase is known as the secretory phase, and you can understand here it is under the control of progesterone. And then you can kind of read all of this. And I'll now go over all of these phases. So the next kind of chart which you see here is showing you all of these phases. So up here you can see this part is the ovarian cycle. This ovarian cycle influences the uterus. So this is the uterine cycle. So look at all of these phases that are mentioned. So let's see what happens. So at the beginning, the hypothalamus releases gonadotropic releasing hormones which act on the anterior pituitary, which secretes FSH and LH. A lot more FSH at the beginning, LH comes later on. This FSH goes and acts on the ovary where you have these primordial follicles, or also known as primary follicles. What the primary follicle consists of is an oocyte with a single layer of cells surrounding it. As these primary follicles begin to mature, we call them growing follicles, this FSH acts on many follicles, so many start growing together. So you can see you have that one single oocyte up here, the cells of the ovary begin to surround and grow in number. So you can see it has lots of cells which are present here. So this is known as the follicular cell. These follicular cells secrete estrogen. Okay? These cells of the ovary, they begin to secrete estrogen. What estrogen does, it goes into the bloodstream and it will act on the endometrium of the uterus. It was shed during the menstrual phase, and then it acts on the endometrium and it causes the endometrium to repair itself. So then the endometrium enters what is known as the proliferative phase. So you can see from this height it's kind of growing larger, the uterine arteries, branches begin to grow, the cells begin to grow, and the endometrial height increases. Okay. Now, under the control of estrogen, you want only one of those follicles to kind of mature. You don't want all of them to mature together and everything to be liberated, because at ovulation, normally only one ovum is liberated. Okay? So what happens, this estrogen, it actually goes back into the bloodstream and it has an inhibitory effect. It has a negative feedback on the gonadotropic hormones and the releasing hormones. So no more FSH is produced. So the follicles are stopped from maturing. Only that one that wins the race continues to win the race. The others all die off. Okay, so that's why there's a lot of cell death that occurs in the ovary. During the later stages of the follicular phase, LH is also is secreted by the pituitary gland. LH acts on that mature follicle and it grows, it acquires fluid, so there's a little cavity which is present up here, so that cavity kind of acts like, provides a little tension. This goes towards the surface of the ovary, and there is kind of a phase where LH amount increases, and that is known as the LH surge, and this secondary oocyte is liberated. So it's sort of sent out into the peritoneal cavity. So here it is liberated at ovulation. Up here you can see these are the cells which are left behind in the uterus. These cells pick up a yellow pigment called lutein, and so it looks yellow in color. So what is left behind is known as corpus luteum. This corpus luteum now begins to secrete progesterone. Progesterone then, and there's a little bit of estrogen also being secreted, but the luteal cells secrete more progesterone than they do estrogen. So now, under the influence of progesterone, the endometrium undergoes what is called the secretory phase. So it's already repaired itself, it continues to grow in height, but now the glands become increasingly tortuous. They begin to store glycogen in them. Glycogen is a source of energy because it can be broken down into glucose. They also begin to secrete mucus and moist substances, so they keep this bed extremely fertile, so that if the ovum, this oocyte which was liberated, if it's fertilized, it will have a good, nice bed, moist, rich, nutritive bed in which to implant. Okay? Suppose fertilization does not occur and this oocyte is thrown out of the body. This corpus luteum shrinks and dies off. So what happens? Progesterone and estrogen are removed from the bloodstream. So now these blood vessels in the endometrium, they undergo spasm, and so ischemia occurs. The endometrium, the surface layers of the endometrium die, and it is shed off in the menstrual cycle, and then the cycle begins all over again. Okay, so this is the phase that occurs. So let's take a look at this.
To understand the various ways that medical science can assist reproduction, it is important to understand how the reproductive system functions in both sexes, because the cause of infertility often lies equally with both men and women. The main players in the female reproductive cycle are the pituitary gland, the ovaries, and the uterus. Their activities are closely coordinated. Each month, one or other ovary releases a single egg, an event known as ovulation. It is brought about by a series of complex interactions between the pituitary gland, the ovaries, and the uterus. The pituitary gland is itself under the control of this small area of the brain known as the hypothalamus. A new menstrual cycle begins when the nerve cells of this center secrete a hormone called gonadotrophin-releasing hormone, GnRH, into the network of blood vessels which surrounds the pituitary gland. Stimulated by pulses of gonadotrophin-releasing hormone, cells in the pituitary gland secrete another hormone, follicle-stimulating hormone, or FSH. FSH travels in the bloodstream, reaching the ovaries. There it stimulates the formation and growth of an ovarian follicle in one or other ovary. The follicle consists of an egg, a number of surrounding cells which secrete estrogen hormones, and fluid. FSH helps the egg to mature and prepares it for release. As the follicle matures, the hypothalamus increases secretion of GnRH. This in turn stimulates the pituitary to secrete a second hormone which acts on the ovary. This is luteinizing hormone, or LH. Toward the middle of the cycle, there is a sudden peak in the blood level of LH. This acts as the trigger for ovulation. Within minutes of its release, the egg is guided by suction through the fringed opening of the outer end of the fallopian tube, starting it on a journey which will take 5 or 6 days as it passes down the tube and finally reaches the cavity of the uterus. Meanwhile, after the follicle ruptures, it is converted into this yellowish body known as the corpus luteum. Cells of the corpus luteum secrete the hormone progesterone, which brings about important changes in the lining of the uterus, preparing it for possible pregnancy. In fact, the lining of the uterus, known as the endometrium, undergoes changes in response to hormone levels during the cycle. In the first half of the cycle, known as the follicular phase, the developing follicle secretes increasing amounts of estrogen hormone, which encourages regeneration of the endometrium. After ovulation, there are important changes in the endometrium, aimed at making it suitable to receive a fertilized egg. These changes are brought about by a secretion of progesterone from the corpus luteum. The secretion of progesterone is maintained for several days, but if the egg is not fertilized in that time, the corpus luteum withers, and falling levels of progesterone and estrogen trigger the shedding of the uterine lining as the menstrual flow. The cycle then starts again. But if the egg is fertilized, no menstruation occurs, as the corpus luteum continues to function, secreting progesterone during the first 3 months of the pregnancy. Thereafter, numerous changes occur to support the developing embryo.
So here in this, if you take a look at this, and this is a typical 28-day cycle. Everybody doesn't have a normal exact 28-day cycle, but if you take 28-day cycle, at about the 14th day, which in this case would be the middle of the cycle, because of that LH surge, as she said, you get ovulation. Ovulation typically always occurs 14 days before the next cycle. So if a woman knows how many days her cycle is, she'll know that 14 days before, that's when ovulation occurs, which means that this phase, which is the secretory phase, it's the more constant phase, because it's only for 14 days, because secretory phase will occur after ovulation, okay? So when women have cycles which are shorter or longer, the phase which is inconstant is the proliferative phase. This is the one which can be shorter or longer. So this is the more inconstant phase. The more constant phase is secretory, because ovulation typically occurs 14 days before the next cycle is about to come. Yes? Okay, good question. Actually, I was going to come to it later. So the question was, where does birth control work? What birth control does is that you start taking it, it's called withdrawal bleeding, because you really don't have this proliferative phase that's going on. So what happens is that you have, let's say bleeding has started, you're finished with it, and you start taking your hormones up here. Birth control pills have a combination of estrogen and progesterone. So already in your blood, the levels of estrogen and progesterone are rather high. So what they do is they go and inhibit gonadotropic hormones, releasing hormones, and FSH and LH. So because these are inhibited, these phases do not occur. So ovulation does not occur. So when ovulation does not occur, you do not have the secretory phase. You get that? So therefore, the bleeding that you have is really not menstrual bleeding. It's called withdrawal bleeding, because you continue for these 21 days and then you withdraw the hormones, and the bleeding that occurs is just because of withdrawal of the hormones. So that's how it occurs, because right from the beginning, the levels are not going slowly like that. You already start with a high level of estrogen and progesterone. So that's how birth control pills work. Okay? Okay, so let's go quickly and look at some questions. So what does the gonadotropic releasing hormone act on? So you must remember that chain of hormone action. Yes, it acts on the pituitary. Gonadotropic releasing is hypothalamus, acts on the pituitary, causing it to produce FSH and LH. What do FSH and LH act on? ovarian follicles. Remember, FSH and LH can only act follicle stimulating hormone, luteinizing hormone. Remember those two phases in the ovary. So they only act on the ovarian follicles, on the ovary. Okay. What are the phases of the uterine cycle? Yes, menstrual, proliferative, and secretory. So that's what the uterus can do. When is the corpus luteum formed? After ovulation, remember, after ovulation, not in the follicular phase. This phase, the luteum is only formed after the oocyte, secondary oocyte has been liberated.